Hi guys, this is Alicia with Good Morning Sunshine. So as you can see, I'm wearing a jacket, so it's now getting cold now. Today we had 4 lectures and we had a BAP class. Luckily we got out an hour early because it was a 4-hour class. And I don't like that class. But anyways, 4 lectures, I think we got into the introduction to bacteria. And then we got into the introduction to clinical pathology, as well as the introduction to parasitology. And there was one more in there. I kind of forgot what it was. I think it was another pathology one. But I am going to start with the intro to bacteria. So it's actually known as bacteriology, our first lecture. This one's a long one, so it may take me a little bit. But all of these lectures are kind of long today, so I'm sorry about that. But at the end of this lecture, we should be able to identify the basic characteristics of prokaryotes, so that's meaning bacteria, to identify the different morphological forms of the bacteria which cause disease in animals, to identify the bacterial ultrastructures and explain their importance in clinical disease, and to explain the difference between gram-positive and gram-negative bacteria, including why this distinction is clinically important. And then to identify the different atmospheric conditions under which bacteria can grow and explain their clinical significance, and to recognize the different mechanisms of bacterial genetic variation and explain how those may contribute to clinical disease in animals. So we had the introduction to bacteria. So microorganisms or microbes comprise a large, diverse group of unicellular organisms that are placed together because they are microscopic in size. So basically, we cannot see them without a microscope. Included in this group are bacteria, fungi, protozoa, viruses, archaea, and some algae. So basically, I'm going to go over bacteria in the different kinds, and then, yeah. So we're going to start with taxonomy. Taxonomy is the science which deals with the study of identifying, grouping, and naming organisms according to their established natural relationship. Taxonomy uses taxonomic units known as taxa, and then singular taxon, to classify things into a taxonomic scheme arranged in a hierarchical structure. This is helpful in microbiology, so that's another name for bacteriology. But basically it's helpful because it accurately identifies organisms, it provides precise names that permits efficient communication, and lastly, they group similar organisms in a way that allows predictions to be made and hypotheses framed with reasonable confidence regarding members of the same group. So then we'll get into biological taxonomy. This is still generally the best known form of taxonomy and is based on natural evolutionary relationships between organisms. So basically the ranks are in order, kingdom or domain. You have the phylum, the class, the order, the family, the genus, and then the species. So with respect to the classification of organisms, the 5-kingdom system has been replaced by the 3-kingdom domain system. So then you have eukarya, archaea, and bacteria. So eukarya are eukaryotes, which are a single cell or multi-celled organism with a nucleus enclosed by a membrane. Archaea are single-celled microorganisms that have an independent evolutionary history from eukarya and bacteria. Archaea and bacteria are quite similar in size and shape, but archaea possesses genes and several metabolic pathways that are more closely related to those of eukaryotes. And then bacteria are prokaryotes that are unicellular organisms which lack a membrane-bound nucleus, and other membrane-bound organelles of eukaryotes thus are smaller and less complex than eukaryotic cells. So classically, bacteria have been further classified into biological taxonomic units based on their phenotype characteristics, including morphology, culture, and staining characteristics, biochemical reactions, cell chemistry, motility, and serology. But more recently, they've been classified based on genotypic characteristics, where methods such as a nucleic acid profiles, multi-locus sequence typing, and 16S rRNA sequences, and are used to classify the bacteria. So bacteria have different characteristics. Some of their characteristics are that they have a rigid cell wall containing a peptid oglycan layer. Nucleus is not bound, like I said, by a nuclear membrane and is usually singular or circular. They do not have a nucleolus, a mitochondria, endoplasmic reticulum, or a Golgi apparatus. They multiply by binary fission, and they exhibit considerable morphologic diversity, but are usually less than 5 units in size. So how do we classify bacteria? Well, by the taxonomy. So bacteria and other organisms are classified further divided into biological taxonomical units. The main divisions we are interested in are genus and species. We classify bacteria using morphology, biochemical reactions, serology, and nucleic acid profiles. So getting into morphology, there are 2 basic morphological shapes of bacteria. There's cocci and rods. So these I worked with at my small animal clinic. But despite their morphological diversity, most bacteria are between 0.5 and 5 units. Cocci, the term coccus, so plural is cocci, can be used to describe any bacterium that has a spherical shape. Like all bacteria, each single coccus bacterium is an entire living organism. However, some species exist in groups of cocci, and the patterns they arrange themselves in are given names and help in identification of the bacterium. So we have diplococci, which is arranged in 2 cell pairs. We have streptococci, which arrange themselves in short or long chains. We have staphylococci, which arrange themselves in clumps or clusters. And then we have sarcina, which arrange themselves in a cuboidal grouping. Of these bacteria, only staphylococci and streptococci have veterinary importance. So then let's get into rods. Bacterial rods, which are sometimes also called bacilli or bacillus, may vary in their shape and size, in which they can include rods, which are straight and cylindrical. We have cocobacilli, which are very short rods. We have curved, which are curved in shape. We have fusiform, which have tapered ends. We have spirochete, which have an elongated, tightly coiled shape, and then we have branching. So here's all the different shapes and arrangements of bacteria. So coccus is up here, and then rods are right there. And then these are all the different forms that I just talked about. So now we're going to get into bacterial ultrastructures. So bacteria are enclosed by a cell envelope, which is made up of a capsule, the cell wall, and the cytoplasmic membrane. The cytoplasm contains various granules, ribosomes, and the nucleoid. Additional structures may include a flagella, pili, also known as fimbrium, spores, and biofilms. So in more detail, the cell envelope includes the cell membrane and the cell wall, plus an outer membrane. Then capsules and slime layer. Capsules or slime layers are amorphous, polymeric, gelatinous materials lying outside the cell wall and are usually described as glycocalyx. Capsules are well-defined structures that are closely adherent to the cell wall, whereas a slime layer is formed when the polymeric material is present as a loose meshwork of fibrils around the cells. Most are composed of polysaccharides, but some capsules may be polypeptides. This chemical makeup makes the bacteria slippery, and heavily encapsulated bacteria have a distinct colony morphology where they look mucoid and glistening. Capsules are not visible in gram stains, but some may stain with special stains, and others are only visible using electron microscopy. They contribute to the virulence of bacteria due to their anti-phagocytic properties. They may help adherence to surfaces, and they also prolong survival in the environment by preventing desiccation. However, they vary chemically and are antigenically diverse, so antibody confers protection only against one specific capsule type, so it's important for host immunity and vaccine development. So more into the cell wall. The cell wall makes up about 20% of the total dry weight of the bacterium and gives the organelles its shape and rigid structure. It also protects the internal structures from mechanical damage and osmotic lysis. It is permeable and only excludes very large molecules. The cell wall confers shape and rigidity, like I said, to the bacterial cell, specifically through peptidoglycan molecules, I think it's peptidoglycan molecules. I don't know. But anyways, these are very large polymers of glycoproteins consisting of alternating subunits of the N-acetyl-D-glucosamine, so G, and the N-acetyl-D-uramic acid. So this alternating structure is cross-linked by short tetrapeptide side chains and peptide cross bridges, making the cell wall very strong. So these bacterial structures are also important that they are targets for the antibacterial molecules such as lysozymes and antibiotics such as penicillin. So the cross-link between the peptide chains is the target site by penicillin, and the linkage between the G and M is the target site for the lysozyme. These molecules act directly on the cell wall to prevent its correct formation, with the resulting bacteria being very vulnerable to lysis when in non-isotonic conditions or due to host defense mechanisms. So we wanted to note that gram-positive bacteria without a cell wall are called protoplasts or L forms, and gram-negatives are called spheroplasts. Within the host, some pathogenic bacteria may still replicate as L forms and cause chronic or persistent infections. Differences in the structure and chemical composition of the cell walls of bacterial species account for variation in their pathogenicity, influence their staining properties, and their susceptibility to enzymes, disinfectants, and antimicrobial drugs. So let's get into the gram stain. So we did this back in undergrad, I believe. I don't remember it much though, so... But anyways, so bacteria are divided into two major groups on the basis of the gram stain. So the gram stain involves the following steps. So it involves a smear, which you put on a glass slide using a sample collected from a case or from a bacterial colony. The bacteria are then fixed, so heat fixed with a lighter. And then next, a basic dye. This would be crystal violet, which is gets it its purple color, is added to the slide, and then it penetrates the cell wall of both gram-positive and negative bacteria. So the next, a mordant, which is an iodine potassium iodide, is added, which combines with the crystal violet that we just put in it inside the bacteria to produce an insoluble large complex. The slide is then rinsed off with water to remove excess dye and the mordant. Next, the slide is decolorized with the ethyl alcohol, where the alcohol dehydrates the peptoglycan of the gram-positive bacteria, making the wall less permeable to the crystal violet iodine complex than the wall of the gram-negative bacteria, which becomes leaky during this process. The di-iodine complex stays inside the gram-positive, but it is washed out of the gram-negative bacteria. The smear is then counterstained with safranin, which is colored red. This penetrates both gram-positive and negative bacteria, but it is only visible in the gram-negative bacteria, as the deep purple of the gram-positive stain masks the pink of the gram-negative stain. So again, there's gram-positive smears and then or stains, and then there's also gram-negatives. So gram-positive bacteria, that means they're stained blue and purple. They have a simpler structure with a relatively thicker, more uniform cell wall, which is composed mainly of peptidoglycan, so that's 90% of the wall, and teichoic acids. Teichoic or lipoteichoic acids are attached to the M residues and a gram-positive. That's all we need to know about gram-positive. Gram-negative bacteria is when it's stained pink or red. So the cell wall of these bacteria are more complex, consisting of an outer membrane and a pyroplasmic space containing a comparatively small amount of the peptoglycan. So that's where they differ. So 10% is the peptoglycan. The outer membrane is an asymmetrical lipid bilayer that also contains proteins. The inner surface is similar to the cytoplasmic membrane, whilst the outer surface is composed of the lipopolysaccharide, or the LPS, or the endotoxin molecules with protein channels called porins. LPS is very important virulence factor of the gram-negative bacteria. So then we're going to get into acid-fast stain. So not all bacteria can be stained with the Gram method. So for example, mycobacteria have a lot of fatty acids, glycolipids, and waxy substances in their cell wall, which makes them difficult to stain. So these bacteria stain with a special procedure called the acid-fast stain, where a strong acid alcohol solution is used as the decolorizer. So now we're going to get into the cytoplasm, ribosomes, and nuclear material. The cytoplasm of bacteria is enclosed by this cytoplasmic membrane. It is essentially aqueous filled, containing nuclear material, ribosomes, nutrients, and other molecules involved in synthesis, cell maintenance, and metabolism. Ribosomes are all protein synthesis with the bacteria takes place on ribosome. Sorry, I'm going to go through these really fast. Plasmids are small circular pieces of DNA which present in the cytoplasm, which are separate from the bacterial genome and are capable of autonomous replication. Phages, so bacteriophages, which are viruses which infect bacteria, are termed bacteriophages. Phages are usually integrated into the bacterial genome or are present as circular DNA in the cytoplasm like plasmids. And then we have additional structures like flagella. Bacteria which possesses flagella are motile. We just needed to know that about them. Then we have pili, also called fimbriae. These are shorter, thinner, and straighter than flagella and are attached to the plasma membrane of mostly gram-negative bacteria. They are composed of a protein monomer called pilin. They are composed, some pili have an adhesion molecule located at the tip of the pilus, which enables these bacteria to adhere to epithelial cells. And then we have spores, which are also called endospores. Some bacteria have the capacity to produce a highly resistant thick-walled spores, which ensures survival in adverse environmental conditions. And then lastly, we have the biofilms. Biofilms are formed when the bacteria switch to the sessile mode of growth. Biofilms start out with the attachment of bacteria to a surface, so like dental plaque, bone implants, catheters, followed by formation of a monolayer of cells. The bacteria inside biofilms communicate with each other through a process called quorum sensing, which this process regulates gene expression in response to fluctuations in cell population densities and thus regulates biofilm thickness. So now we're going to get into bacterial growth. So small colonies equal slow growing, whereas big colonies equal fast growing. Appropriate conditions of moisture, pH, temperature, osmotic pressure, atmospheric, and nutrients are required for bacterial growth. Bacteria increase in number by binary fission, like I said earlier, where the generation time is the length of time required for a single bacterial cell to yield 2 daughter cells and is influenced by both genetic and nutritional factors. Temperature. So most pathogenic bacteria can grow on nutrient media at 37 degrees Celsius, which is around 98.6 degrees Fahrenheit, close to the normal body temperature. Although this is the optimal temperature for growth, most are capable of growing in temperatures ranging from 20 degrees Celsius to 45 degrees Celsius. So Listeria, which are cold tolerant and will grow at refrigerated temperatures of 4 degrees Celsius, which is used to help isolate these bacteria. So getting into pH and tonicity, optimal pH is neutral to alkaline, and optimal tonicity is isotonic to hypotonic. So then we have a few atmospheric requirements. Based on their preference for particular levels of oxygen, bacteria can be assigned to 4 main groups. So first group being strict or obligate aerobes. These bacteria utilize metabolic pathways in which oxygen is the final terminal electron acceptor, respiratory catabolism. Therefore, they can require oxygen to grow and can be incubated in air. Strictly aerobic pathogens are not common, some occurring in the mucosa of the upper respiratory tract. So then second, we have the strict or obligate anaerobes. In contrast, anaerobic bacteria are unable to grow in an atmosphere containing oxygen, and often even small amounts of oxygen is toxic. These organisms use fermentative pathways in which organic compounds serve as the final electron acceptors. And then thirdly, we have the facultative anaerobes. Facultative anaerobes are bacteria which have the ability to grow under both aerobic and anaerobic conditions, but better with oxygen. Many of the major pathogens are facultative anaerobes, where many are opportunistic infections, as they are found as normal flora of the skin and GIT, the gastrointestinal tract. Then lastly, the fourth thing, we have the microaerophiles. The microaerophilic bacteria require reduced oxygen concentrations for growth, concentrations lower than in air, so this would equal about 20% oxygen. Many also require increased CO2. So now we're going to get into bacterial genetics and genetic variation. So plasmids, bacteriophages, and transposable elements may contribute to additional genetic information, some of which may influence phenotypic expression. So basis of bacterial variants, we have the phenotypic variants and the genotypic variants. Genotypic variants include mutation, which is the change in chromosomal DNA. Then we have recombination, which is the exchange of DNA between bacteria. And then we have transposition, which is the relocation of parts of DNA in the genome mediated by transposable elements. And then recombination also includes transduction, which is the phage-mediated transfer. We have conjugation, which is the plasma-mediated transfer. And then we have transformation, which is the reuptake of naked segment of DNA. So then we have mechanisms contributing to genetic variation. So genetic variation may occur following either mutation, in which a change occurs in the nucleotide sequence of a gene, or by recombination, in which new groups of genes are introduced into the genome or into the cytoplasm as separate genetic elements. We have mutation, which is a stable and a heritable alteration in a bacterial genome, which is termed a mutation. Then we have recombination, which occurs when sequences of DNA from two separate sources are integrated. And then again, we have transduction, which are viruses which infect bacteria, are called, like I said, bacteriophages. And then we have conjugation, which involves the transfer of genetic material through the action of specialized fertility plasmids. And then transformation is the process which involves the transfer of genes in a free segment of chromosomal DNA from a lysid donor bacterium to a competent recipient. Transposition results from genetic elements called transposons and or integrons. So that was a lot of stuff for one lecture. That wasn't even all of it. And then now I'm going to talk about clinical pathology, so red blood cell mass and indices. So we're going to start with erythrogram and anemia. I think we just got it through erythrogram today, though. So lecture overview, we're going to interpret erythrogram results, and then I don't think we got into the introduction of anemia, but that's going to be part of it too. So red blood cell mass has, there's like 4 different, I think, categories. We have analytes, which is the PCV is not, so the packed cell volume is not from an analyzer, whereas the hematocrit is from the analyzers where air can be produced. So red blood cells have a number, and then hemoglobin we get from the amount. So definitions, erythrocytosis is if the red cell mass is an increase, so above the reference interval. Most common cause is dehydration. And then we have anemia, which is when the hematocrit is below the reference interval, so the red cell mass is reduced. So variables would be the red blood cell concentration and hydration status. So severity is when a PCV equals 23%. So a dog is usually between 23 and 33%, which that would be moderate, and a cat is between 23 and 26, which is moderate. A dog's perfect PCV count is 45, whereas a cat is 35. So then we have the red blood cell mass artifacts. This is where we're introducing air and so we want to analyze as soon as possible the PCVs. because if not, we're introducing air. So the PCV can be messed up by sample storage, the hematocrit. So all these, both the PCV and the HCT will go up if there's air. The red blood cells, their sample storage, if hemolyzed, the red cell volume would go down, and the red blood count, agglutination, so the red cells start to stick together, so the red cell thus would go down as well. Hemoglobin in lipemia will give us a high value for hemoglobin. So if we let the tube sit, so the PCV tubes sit too long, we will have skewed results. So then we got into reticulocytes and the polychromatophils, which are young erythrocytes. So above the reference interval, then it's a regenerative anemia, so blood loss or a hemolytic event took place. So anything above 1 in reticulocyte percentage, that means they have a lot of polychromatophils. But non-regenerative anemia, which means it's below the reference interval, so anything lower than 80,000 in a dog is usually non-regenerative. So again, getting into analytes, percent reticulites, so we need an absolute number of red blood cells instead of just the percentage. So we usually look at the absolute number of reticulocytes when we're looking at analysis. So definitions, the reticulocytosis is the same as polychromasia. So increased polychromasia means the increased number of reticulites. So that means increased polychromatophils. So 1% being the reference interval, so 50 to 80,000 in dogs, and in cats it's 60,000 to 70,000. So variables include reticulocyte concentration and the duration and severity of anemia. So the uses is bone marrow health and RBC regeneration, so red blood cells hyperplasia. So then we have nucleated red blood cells. So we see a low number of these in anemia. So again, getting into analytes, so it's nucleated red blood cells divided by 100 white blood cells. Definitions, so metaerythrocytosis, which is the last stage before the polychromatophils. Variables include nucleated red blood cell concentration and white blood cell concentration. So its uses are for bone marrow health and splenic health. So when the spleen isn't working, there's a more number of nucleated red cells. So there's appropriate metaerythrocytosis. So if you see more than 5 nucleated reds, ask yourself if this is appropriate or not. So we have erythroid hyperplasia, which would be the appropriate one, but then we have inappropriate metaerythrocytosis. This has to do with bone marrow, so leukemia, fracture, hypoxia, and then splenic disease. So if we see a lot of nucleated reds in the vascular space, this is not good, so that means no spleen or cancer. And then we have the mean corpuscular volume, so the MCV definition. So macrocytic means it's too big, normocytic means it's normal, and then we have microcytic, which means it's too small. Variables include erythrocyte and reticulocyte size, and the uses are macrocytosis if red cells are too big. Microcytosis is if red cells are too small, so during their maturation process, they're losing capacity to produce hemoglobin, so they just keep dividing, which thus gives them the smaller size. Polychromatophils are too big, so they're more immature. So microcytosis, the artifact being short draw, so excess EDTA, which equals a small red blood cell. And then we have hyponatremia, which is a small red blood cell. So physiologically, young animals less than a year, the red cells are smaller at this age, and Asian dog breeds such as the Akita, the Shiba Inu, and the Shar Pei have smaller cells because of their breed. Pathologically, there's iron deficiency, so prototypical cause is microcytosis. So liver disease also, so failure or portosystemic shunt will cause the microcytosis. And then we have macrocytosis, so the artifact being red blood cell agglutination, which means, again, they're sticking together, becoming a large red blood cell. And then we have sample storage, which equals the large red blood cell, and then hypernatremia, which also produces a large red blood cell. So physiologically, this is a normal finding in poodles. Pathologically, the reticulocytosis is a regenerative anemia. We have FELV infection, which interferes with nuclear division. And then we have folate cabalin deficiency, which these vitamins are critical for nuclear division, so the red blood cells then stay big. Then we have the mean corpuscle hemoglobin concentration, so the MCHC. Definition, we have hyperchromic, normalchromic, and hypochromic regeneration. Variables being hemoglobin and HCT, so hematocrit, and then we have erythrocyte and reticulocyte hemoglobin content. Its uses are for hyperchromasia, so that doesn't really happen. But then we have hypochromasia, which occurs in association with iron deficiency. So hypochromasia, its artifact being sample storage, which produces a large red blood cell. And then we have hyperatremia, which is a large red blood cell, like I said earlier. Pathologically, reticulocytosis means an increase in red blood cell volume. Iron deficiency, you have a decrease in red blood cell volume. Liver disease, again, you have failure in the portosystemic shunt, and then the deficiency in the vitamins that I told you. With hyperchromasia, its artifact being leukemia, hemolysis, and short draws, which means a small red blood count, and then hypoatremia and Heinz body. Anemia. Oh, we didn't get into anemia, so I'm not going to talk about anemia yet. So that was it for clinical pathology 1. And then we got into animal pathology. So our objectives for animal pathology 1 is the student will be able to define pathology and explain its relationship to clinical medicine, describe basic terms related to pathology, and describe intrinsic and extrinsic factors that cause disease. So pathology is not disease. Pathology is the science that analyzes the molecular, functional, and structural response of cells and tissues to an aggression, aiming to understand the process of disease and to reach a diagnosis. So basically, it is a science that studies disease. It is not a disease itself. So there's different levels of complexity. So it goes from the populations to the individuals, to organ systems, to tissues, to cells, to organs, to genes, to molecules. So why is veterinary pathology important? Well, veterinary pathologists advance animal and human health through 1, disease diagnosis in companion food producing and zoo wildlife animals. It is central to the discipline of veterinary pathology and is essential to the health of our pets and our food supply, as well as the conservation of wildlife species. Veterinary pathologists serve as key members of drug discovery and development teams, which aim to bring safe and effective drugs to the market for both human and animal use. Because veterinary pathologists have a deep understanding of the diseases of multiple species, they are uniquely qualified to perform research that advances our knowledge of disease pathogenesis and leads to development of new methods to treat and prevent disease. So examples of contributions of veterinary pathologists. They recognized that the West Nile virus invaded North America. They conducted experiments on the space shuttle. They responded to environmental disasters such as the nuclear accident and the Exxon Valdez oil spill. They also performed pioneering stem cell research. So we do have history of pathology. I'm not really going to go over that with you because it's a lot of information. So we'll skip that part. But so areas of veterinary pathology includes virology, parasitology, bacteriology, and a lot more others. But pathology is most useful when it's used as a diagnostic tool. The descriptions and interpretations identify or ideally help the observer to get back to an etiology or pathogenesis, and it helps to keep in mind a general list of mechanisms of the disease. So definitions in pathology. So we have anatomy, which is the shape and structure of an organism. We have anatomic pathology, which is the morphological alterations. We have pathogenesis, which is the mechanisms of disease. We have clinical signs, which diseases manifestation in the animal alive. So animals don't have symptoms, they just have clinical signs. The diagnosis is the medical ability of recognizing lesions in a live or dead animal and understanding the etiology and pathogenesis and establishing recommendations for treatment and control and prevention of a disease. So we have necropsy, which is a post-mortem examination. We have lesion, which is abnormal tissue damage. We have etiology, which is the cause of disease, which include different factors such as predisposing factors, which would be cold and overcrowding. And then we have determinant factors, which include infectious agents and deficiencies. So here I'm going to show you this diagram here so I can stop talking for a minute. But here we have disease etiological factors, so we have predisposing factors, we have intrinsic and extrinsic. So intrinsic includes species, breed, age, sex, color, and idiosyncrasy, whereas extrinsic includes temperature, so tropical, dry, warm, cold, or polar, and then we have nutrition, so in excess or deficiency. And then another determinant factor is intrinsic, which includes genetic abnormalities like autosomal linked to sex, dominant or recessive. And then determinant factors also include extrinsic, which include physical, biological, and chemical. Biological includes prions, virus, bacteria, fungi, parasites, and humans. Whereas physical includes mechanical, thermal, atmospheric pressure, and actinic. And then chemical includes exogenous, which includes toxins, poisons, drugs, medicines, and the endogenous includes metabolites and free radicals. So starting with the intrinsic predisposing factors of disease, like I said, species, breed, age, sex, color, and idiosyncrasy. We're going to start with species. So this is myxomatosis, so swelling of the eye in a rabbit. And then here we have a verminous aneurysm, so parasites in the mesenteric arteries in a horse. So this is the order of a horse right here. Then breed, so we have mastitis in this cow right here. And then boxers are predisposed to different tumors as examples of breeds, intrinsic factors. And then we have age, so young dogs get parvovirus, old dogs get benign prostatic hyperplasia. Young calves have different causes of diarrhea. They have salmonella, E. coli, rhabdovirus, coronavirus, or cryptosporidium. And then sex being another intrinsic factor. Females are predisposed to pyometra and mastitis, whereas males are predisposed to testicular tumors. And then getting into color, we have squamous cell carcinoma of digmig-pendant areas in white. And then now getting into extrinsic predisposing factors. So food excess, we have lamitinus, which is carbohydrate overload treatment, which includes fever, colic, and sepsis and other causes. We have the gastric impaction in horses. And then we also have obesity. And then extrinsic predisposing factor of lack of food. So we accumulate fat, starting with bone marrow to pericardial groove, which is the heart, the perirenal area, which is the kidneys, the abdomen, and then our subcutis. We remove fat. So when we start exercising, the fat that comes off first is our subcutis, and then our abdomen, and then down the line backwards. So extrinsic predisposing factor, lack of nutrients, we have thiamine, which is vitamin B1 deficiency. Here we have iodine deficiency, we have zinc deficiency here, we have calcium phosphorus imbalance, which means rickets, and then we have osteomalacy. So determinant factor or cause that makes something happen or leads directly to disease. So then we have extrinsic determinant factors, which are mechanical. We have abrasion, which is skin damage with loss of epidermis and portion of dermis. So this is a superficial wound here at the bottom. And then we have lacerations, which is deep cut or tear in skin of the flesh, which we have here. Then a puncture is penetrating wound caused by a sharp object, which is here. An incision. Oh, I touched my screen. Hold on. Let me see. And then incision is what we do. So a wound created by a sharp tool, which would be a scalpel in our defense. And then we have perforations, which is a hole that develops through the wall of a body organ. So this is where we have exploratories when a dog eats something and we have to get that object out. So these are foreign bodies here. We have rupture, which is a break or tear in any organ or soft tissue. And then we have a fracture, which is the cracking or breaking of a hard object or material, which is usually the breaking of a bone. So then we have extrinsic determinant factors, actinic radiation, which means UV light, and which equals mutations. And then we have extrinsic determinant factors, which are chemicals. So we have the endogenous toxicants, which includes your uremia and burns and gangrene. And then we have the exogenous toxicants, which includes the mycotoxins, the rodenticides, insecticides, and toxic plants. So then we have extrinsic determinant factors, which are biologic as well. So we have foot and mouth disease in cows, which we need to rule out first before we do anything. And then we'll check for vesicular stomatitis or IBR or MCF, BTV, EHFD, or BVD. So not only DNA or RNA agents, so mad cow disease, so bovine spongiform encephalopathy in England due to prions, infecting protein particles without nucleic acids, very resistant to the common mechanism pathologic inactivation, and they cross the species barrier, so ovine scrappy and Creutzfeldt Jakob disease in humans. So then we have post-mortem examinations. So these are tools of the veterinary pathologist. So here we start with a horse, then we cut it open, and then we see the abdominal right there. And then we have virulent Newcastle disease, which was in fighting cocks, also known as VND, he wanted to show us. And then another tool is histopathy. Another tool is electron microscopy. And then we have molecular techniques and immunohistochemistry. And then we have a suggested reading. So that was animal pathology. And I think that was it. Did I go through everything? I don't remember. So it was animal pathology. Let me look. Because I feel like I was supposed to have one more lecture. What did I tell you guys? I think I do. Parasitology, right? I don't know why that document went away. So let me find my parasitology one. Here we go. So we have the introduction to parasitology now. So objectives here to provide background and vocabulary in parasitology. So we have to be prepared for later courses. We have to understand the literature and appreciate future developments. So what is a parasite? It is an organism that lives in or another living organism, so that being the host, obtaining from it part or all of its nutrients. So that's a wide definition. It can cause harm to the host, some degree of adaptive structural modification, and parasites only include those organisms that are eukaryotes. So we have the protozoa, we have the arthropods, we have the platyhelminths, and the nematoda. So protozoa include examples are Giardia, the trypanosums, and the coccidia. The arthropods include ticks, mites, fleas, and mosquitoes. The platyhelminths includes tapeworms and flukes, and the nematoda include roundworms and heartworms. So parasitology, parasites can either have a direct life cycle, which means one host, or they can have an indirect life cycle, which means two or more hosts. So classification of hosts, definitive hosts are hosts in which sexual reproduction takes place, also known as the final host. We have intermediate hosts, which are needed. They are hosts in which only immature stages grow and develop. They may or may not occur asexual reproduction. And then we have the paratenic host, which is just an accessory. It's not required. It's just used for transport, and there is no development of the parasite. So this host is not required to complete the life cycle, but help the parasite to get the host. So there's no parasitic development of any kind that takes place, and it's also known as a transport host. So more general terms, not limited to parasites, but you have a normal host, which is an animal recognized as part of the normal life cycle. We have an abnormal or aberrant host, which get disease but aren't going to transmit it. It could be due to accidental transmission. So the animal not normally used as a host is often not always associated with disease, and the animal could be an abnormal, definitive, intermediate, or transport host, depending on which stage of the parasite life cycle is present. And then we have the reservoir, which can get affected, but usually doesn't get the disease. The host serves as a source of infection for other animals. And then we have the vector, which is usually an invertebrate, usually an arthropod that determines infectious disease. So we have a biological vector, which is a parasite, which the parasite develops in the vector. And we have a mechanical vector, like a contaminated instrument. So then we have the host-parasite relationship. So the location in the host is either an endoparasite, which means inside the host, or an ectoparasite, which means outside the host, so an external parasite. So then we have infection, which means endo, which means inside. So it's which means just the presence of the parasite. So it just means this is in your body, basically. It knows the animal. It knows animal is sick during infection, but versus infestation, so that's ecto, that's parasites on the outside, and we don't know if the animal is sick or not in this case. So we have localization in tissue, so we have extracellular and intracellular, and then we have reproductive behavior in the final host. So we have microparasites, which can multiply inside the host, and then we have macroparasites, which cannot multiply inside the host. So we have infective versus infectious. So these terms are often used synonymously, but in parasitology they have slightly different meanings. So infectious is caused or capable of being communicated by infection, whereas infective, the parasite stage capable, which is the parasite stage capable of infect the host. So many parasites are not able to cause infection until after some period of development in the environment. So example of this would be when an animal eats the infected bacteria. This is the stage that infects the host. So that would be the infective stage. And then we have a pre-patent period. So another example, so when the cow eats the spore-laden oocyst, there is a period of time that is required following infection of the defendant host to reach reproductive maturity, so sexual reproduction. So the period before the production of the egg cyst, so you won't see the eggs and feces for a little bit after the host is infected. This is due to the pre-patent period. So this is a term used with internal parasites, mainly macroparasites. It's the time required following infection of the definitive host to reach, again, reproduction maturity. So it's not the same as the generation time. So another pre-patent period example, so in heartworm, it is 6 months long. So at the time an infection becomes patent, important because often parasites can be diagnosed when infection is patent. Generation time would be pre-patent period 6 months plus the time to develop the infective stage in the mosquito, so 2 weeks minimum. So the patency period is 5 to 7 years. So then we have a host-parasite relationship. When animals can be affected but not get the disease, will an exposed animal become affected? Parasites usually require appropriate stimulus to recognize potential host. Intercellular parasites need specific receptors on the cells of appropriate host, and GI helminths often need specific cues like pH and bile. So not all of the people that get, or animals that get exposed become ultimately infected, if that makes sense. So they can be infected but also not get the disease if they don't have the correct stimuli to do that. What determines outcomes of host-parasite relationships? So will an infected animal become ill? So again, infection, infestation versus disease. Parasite usually requires appropriate stimulus, like I said, to recognize potential hosts. So host response is either innate immunity or adaptive immunity. Normal immunity typically limits but does not eliminate parasite infection. So it limits reinfection, reduces growth and reproduction of parasites, so often full expression of immunity requires presence of active infection. So sometimes younger animals don't have a developed immune system, so sometimes they will get a disease worse because of their not developed immune system. So how do parasites cause disease? Well, pathogenesis depends on 3 factors: the status of the host defenses, the number of parasites present, and the pathogenesis of these parasites. In general, pathology from parasitic infection may be described as the result of 1. Parasite-induced trauma to cells, tissues, and organs. 2. Changes in cellular growth patterns. 3. Interference with host nutrient acquisition. 4. Toxins released by the parasites. And 5. The host immune response infection. So what happens when animals get parasites? Well, as the number of parasites keeps going up, they become more awful or ultimately become dead. So the parasite populations in animals are aggregated, so over dispersed. So some have many, whereas some have many, and then some have very few parasites. So few animals should have more, but there's more animals here that have few parasites, whereas there's a few that have a lot. So that's what it means by aggregated. So a small amount have a lot of parasites, whereas overall, many of them have a small amount of parasites. So aggregated parasite populations, so individual hosts vary in susceptibility. So temporary factors include age, concurrent diseases, and nutrition, genetic difference in individual susceptibility. All other things equal, most normal animals maintain low to moderate parasite burdens, so a limited number of animals have high susceptibility. A small number of animals contributes disproportionately to parasite transmission. So remember that infection does not equal disease. So then we have the 70-30 rule. So the top 30% of animals, so the 3 sheep in this case, have 70% of the parasite population. So like I said, very few have many of the parasites, and then the rest have very little parasites. So that's how it works. So what is the veterinarian's role? Well, our role is understanding the significance of the infection, the prevention of infection, integrated parasite management, diagnosis, and treatment, and of course, but technology changes diagnosis and role of the internet. So diagnosis of parasites, we have morphological, microscopic examination of blood, tissues, and feces. Gross inspection of tissues or feces remains the most common way to detect parasites, especially multicellular parasites. And then we have immunological assays, which are the antigen or antibody tests used mostly with protozoan parasites. And then we have molecular tests, which include the PCR. And then we have parasitological principles. Parasitic infection is not synonymous with parasitic disease. Immunity often presents disease, but not infection infestation. Young animals are usually more susceptible to parasitic infection and disease than adults, so that's the poor baby rule. And then parasites that have stages that must develop and survive in the environment are at the mercy of the weather, so temperature and moisture. And then if you want to kill a parasite in the environment, you have to desiccate it. So that is the end of lectures today. I know almost an hour. I tried to rush through them, so I'm sorry if they're really boring. It's a lot of information. Dealing with threats is supposed to be the hardest class, so it's gonna be great. But I love you guys, and I thank you guys for watching always. Even if you don't really watch them, it's just a tool for me to go over the information again, and then also be able to watch it back when I'm studying for the big NAVLE. But yeah, I love you guys, and I'll talk to you guys tomorrow.